Sex-linked genes are genes located on the sex chromosomes. These genes have different inheritance patterns than the other chromosomes in the nucleus, which are the autosomes. For sex-linked traits, we see parents producing offspring with different phenotype ratios divided by sex. Why is that? Let's investigate further.
Here we will see two flies. One is a female XX with red eyes.
The other is a male XY with white eyes.
In Drosophila melanogaster, a sex-linked trait that is easily identifiable is eye color. The wild-type fly has red eyes. Flies can have many different eye colors, including brown or a brighter shade of red. But it is important to note that many genes contributing ultimately to eye color phenotype are not actually sex-linked, such as the brown and scarlet genes, which we renamed Princeton and Harvard in class for fun. However, the white eye gene, which is mutated in me to give rise to my white eye phenotype, is sex-linked.
Say these two flies were to mate and produce offspring. What would their offspring look like? An easy way to predict this is through a Punnett square, which shows the genetic contribution of each parent during fertilization and the expected genotypes of their offsprings. A female fly will contribute her X chromosomes. As we said, the eye color gene is sex-linked and it is located on the X chromosome. We happen to know that this female is homozygous for the wild-type gene, therefore she will contribute an X with a wild-type gene to each of her potential offspring. It is a 50% chance that the male will pass on an X chromosome or a Y to his offspring. This is where we start to see unique ratios. Because the male only has one X chromosome, he only has one copy of the white gene to contribute. This is hemizygosity, where an individual has only one allele copy in the genome, so recessive mutations, such as the white mutation, are more frequently observed in males that have only one X chromosome.
This is also the case for color blindness in humans. It is more often that the XY individuals are colorblind than XX individuals. For any daughters produced in this cross, both the male and the female will contribute an X each, which means that all of our daughters are heterozygotes for the X-linked white gene. We see that in this cross, the male has the mutant gene on the X chromosome, while the female carries the wild-type allele on both of her X's. However, the father must contribute his Y chromosome to his sons, so the mother is solely responsible for the X-linked traits in her sons. In this case, this pairing will have all wild-type sons. Thus, all of the progeny, no matter the sex, will be phenotypically wild-type.
Now let's look at a reciprocal cross. A reciprocal cross is a secondary cross that also examines sex-linked traits. Between the first cross and this cross, the phenotypes of the sexes are switched. Before we had a red-eyed female and a white-eyed male, and now we have a red-eyed male and a white-eyed female. What will their crosses look like? The white-eyed female has two mutant genes to contribute to all of her potential offspring, so the deciding factor in the phenotype of the offspring is the father's contribution. His X chromosome will go to his daughters, and this X chromosome has the wild-type gene. In this cross, the F1 daughters will again be heterozygous and have the wild-type phenotype. However, look at the males. The father contributes the Y chromosome, so their only copy of the white gene comes from the mother, and in this case the gene is mutated. Therefore, this cross will only produce white-eyed sons.
To review, here are the F1 phenotypes that we've amassed so far. Also, we want to acknowledge that sometimes in this world we see things that we do not expect.
Here is a Punnett square for my parents. If there should be a 0% chance of females with white eyes coming out of this cross, what could have caused me? Usually, variability and unexpected results like this comes from using non-virgin female flies. Female flies can be inseminated by one fly and hold on to the semen for weeks at a time, even when separated into mating crosses. In this case, we would expect that the mother of this fly had been previously inseminated by a white-eyed male fly, which would produce the white-eyed daughter with two mutant genes.
It is important to ensure good practice in the lab to avoid this kind of variability because it makes it so much harder to understand what is going on genetically when we have unaccounted for variability like this. Therefore, virgin females are essential in genetics.